Before we proceed to how phloem translocate sugar, let's see the evidences that prove sugars are transported by phloem.
The evidences are, first, wringing the bark of a tree always causes the tree to die. Before it dies, it can be observed that sugar and other organic compounds accumulated at the upper part of the ring like in figure 1. This is because the wringing process removed the phloem, and solutes cannot pass across it into the root. The second evidence is, staining the organic substances also stain the contents of the phloem. This indicates that phloem is involved in transport of organic solutes.
There are hypotheses stated about the mechanism of phloem translocation.
Pressure flow hypothesis, cytoplasmic streaming hypothesis, electroosmosis hypothesis, and peristaltic wave hypothesis. But we'll only discuss on the first two hypotheses.
This apparatus of beakers and funnels simulates the flow of a sucrose solution in the phloem of a plant. The funnels and connecting tube represent a sieve tube of the phloem. Differentially permeable membranes cap the funnels at the source and sink ends, allowing water but not sucrose to cross. Let's consider an experiment in which each funnel contains the same low concentration of sucrose. Water diffuses by osmosis into both funnels equally. Since neither funnel takes on more water, there is no net movement of the solution in the tube. As internal pressure builds, water no longer enters either funnel. Similarly, in a phloem sieve tube that contains equally low concentrations of sucrose along its length, a small amount of water enters the phloem by osmosis. However, the water enters equally along the length of the sieve tube, so there is no net movement of sucrose. In an experiment in which one funnel contains a high concentration of sucrose and the other contains a low concentration, water initially diffuses into both funnels, but soon only diffuses into the source end. As water flows into the source end, the solution is pushed through the tube and water is forced out the other end. Now consider a sieve tube that contains a high concentration of sucrose at one end and a low concentration at the other. Water initially flows by osmosis into both ends, but flows much more abundantly into the source end. This incoming water at the source end pushes the sucrose solution through the tube. vascular system of plants has two tissues called xylem and phloem. Xylem transports water and minerals, while phloem transports a variety of dissolved substances, including sugars and amino acids, throughout the plant. Water in the xylem always moves up in the direction from the roots to the leaves. At the leaves, water evaporates. This evaporation, called transpiration, creates tension at the top of the water column in the xylem and causes water to be drawn upward from the roots. Unlike in the xylem, the solution in the phloem can flow up or down the plant. The direction depends on the concentration of solutes in the phloem. A model, called the pressure flow model, describes how sucrose concentrations determine the direction of the flow. Two types of cells, source cells and sink cells, play roles in the transport of solutions in the phloem. Source cells produce sucrose and load it into the phloem. Source cells are typically photosynthetic leaf cells, but they can also be other cell types. For example, root cells with large stores of carbohydrates can act as source cells by releasing these carbohydrates into the phloem. Sink cells are any cells that unload sucrose from the phloem. Sink cells are found throughout the plant, but are abundant in roots or developing fruits and shoot tips. Sink cells typically cannot meet their own needs for carbohydrates and must import them from the phloem. The fluid begins to flow when source cells pump sucrose into the phloem. As the concentration of sucrose in the phloem increases, water enters the sieve tubes by osmosis. The water enters from any surrounding tissues, such as the xylem, that have lower solute concentrations than the phloem. As water flows into the sieve tubes, the turgor pressure within the sieve tubes increases. This pressure forces the sugar solution through the sieve tubes to regions of lower pressure. The solution passes by sink cells, and these cells take up sucrose. As the sucrose concentration decreases, water flows by osmosis out of the phloem to regions of higher solute concentrations. The loss of water, in turn, decreases the pressure in the phloem and pulls fluid from other, higher pressure regions of the phloem.
However, there are some scientists against this hypothesis because this hypothesis does not require a sieve tube to have sieve plates and cytoplasm. Sieve plate is a hindrance to mass flow. It cannot explain the fast reverse flow from the root to the leaves. However, recent works show that older sieve tubes have little or no cytoplasm and the sieve plates are required to prevent the tubes from bulging too much. Generally, the pressure flow hypothesis is accepted as a major contribution to the translocation of organic substances, if not the only way.
Initiating system launch. System 1 loaded.
none of the existing theories to explain translocation is entirely satisfactory. The mechanism of phloem translocation remains one of the unsolved problems of plant physiology. Physiological evidence does not clearly disprove or support any hypothesis.